Students, today we'll start with structures of or components of a cell. Right? So, first component we are going to discuss is cell wall. Now, what is cell wall? Cell wall is non-living and rigid structure. If we compare cell wall with plasma membrane, then plasma membrane is a living component of the cell, while cell wall is non-living component of the cell. Right? It is rigid component or rigid structure. Structure means it will provide structural support. Structural support to the cell, right? Most importantly, now what is cell wall? Cell wall is outer covering of the cell. Is it the outer covering of cell of all the type of cell? No. In number one, plant cell wall is the outer covering. Number two, fungal cell or fungi. Then bacteria and some of the protist. In these organisms, cell is covered by a rigid non-living structure, that is cell wall. Now, if we compare component of all these cells, suppose if we are talking about plant cell, then cell wall is made up of cellulose, hemicellulose, pectin, and some proteins, right? If we consider cell wall of fungi, the main component of cell wall of fungi is chitin. It is very important for your NEET examination. It has been asked many times that cell wall of fungi is made up of chitin. Cell wall of plant is made up of cellulose, hemicellulose, pectin, and some protein, or we can say lignin, right? Next is bacterial cell wall. Bacterial cell wall is made up of peptidoglycan. We have already discussed about it in prokaryotic cell, right? And protists are similar to plant cells, so cell wall of protist will be similar to cell wall of plant, that is cellulose, hemicellulose, pectin, and some proteins, right? Now there is one more important point: cell wall of algae. Component of cell wall of algae. Algal cell wall is made up of cellulose, that is the main component. Other than cellulose, they have galactans, mannans, and minerals like calcium carbonate. So you have to remember components of the cell wall of plant cell, which is cellulose, hemicellulose, pectin, and protein. Fungal cell wall, main component is chitin. Bacterial cell wall, peptidoglycan. Algal cell wall, cellulose, galactans, mannans. There is one spelling error. Just make it correct. Mannans, right? Galactans, mannans, minerals like calcium carbonate. These are the component of the cell wall, right? Now we'll be discussing about type of cell wall, or we can say formation of cell wall. How cell wall is formed? Suppose we have a plant cell. This is the plant cell. At the time of division, two nucleus are formed, right? Two nucleus are formed at the middle of the cell. There is deposition of Golgi bodies, right? These Golgi bodies will form a layer of pectate. That is a layer of pectate. This layer of pectate is middle lamella. Or we can say, other than middle lamella, we can also call it cell plate. And this cell plate is termed as precursor of cell wall. Right? Now, this pectate will start growing towards the end. Now, suppose there is deposition of middle lamella, that is middle lamella. Now, this middle lamella will form a layer on both the sides, toward this side and this side. Both the side, a layer will be formed, and that layer is termed as, this is primary cell wall. Now, what is primary cell wall and what are the features of primary cell wall? This primary cell wall, so first layer of cell wall, let's do it here. First layer of cell wall is primary cell wall, right? Which is formed by middle lamella, right? It is thin, elastic. As the cell is formed, this is the first layer to be formed, which is, that is why it is known as primary cell wall. So it is thin, it is elastic, it is composed of cellulose, hemicellulose, and pectin. Right? Lignin is not deposited. Lignin will be present in secondary cell wall because concentration of lignin or component of lignin or deposition of lignin will make the cell wall rigid and hard. And we are saying that primary cell wall is thin and elastic. So elasticity is provided by pectin and rigidity is provided by lignin. Right? Next important note about primary cell wall is it gradually diminish as the cell matures. What does it mean? Suppose the cell is getting mature at the time of maturation, what will happen? This is a cell. These are two nucleus. First layer of pectin is middle lamella. This is middle lamella. On both the side, primary cell wall is formed. On both the side, primary cell wall is formed. Now, as the cells start maturing at the time of maturation, it will form secondary cell wall. This primary cell wall will form secondary cell wall towards the inner side. That is the inner side. That means towards the cytoplasm. This layer is known as secondary cell wall. So we can also ask question like this, that secondary cell wall is towards the cytoplasm. You have to draw all the diagrams. It is very important, towards the cytoplasm. And when we talk about primary cell wall, it is towards the middle lamella, that is outer side, right? Middle lamella. So as the cell matures, deposition of secondary cell wall will increase and primary cell wall will gradually get diminished. Diminish means it will disappear. That is why we call that primary cell wall is capable of growth. Right? Means this is elastic, thin, it can grow, it can expand, it can be able to perform major function like cell division. So there is one more important point, mainly present in meristematic tissues. In meristematic tissue, secondary cell wall is completely absent. Secondary cell wall is not present in meristematic tissue. Second type of cell wall is secondary cell wall. If primary cell wall was thin and elastic, then secondary cell wall is thick and rigid. Right? Thick means it is made up of three layers: S1, S2, and S3. Component of secondary cell wall, like for primary cell wall, we've just discussed that components are cellulose, hemicellulose, and pectin. Here, the component is cellulose, hemicellulose, pectin, and lignin deposition start in these cells. Cellulose, hemicellulose, pectin. These are the main component, but lignin deposition starts in secondary cell wall. That is why secondary cell wall as compared to primary cell wall is rigid. Most important point, this secondary cell wall is absent in meristematic tissue. We'll discuss about this topic, that why it is absent in meristematic tissue, but in anatomy portion, not here. Okay. Now there's one more important layer, third type of cell wall, that is tertiary cell wall. Tertiary cell wall is not present in all the cells, right? It is present only in tracheids of gymnosperm. It is not very important to learn. It is just an exception that in tracheids of gymnosperm, there is an additional tertiary cell wall is also present, and the component is tertiary cell wall is hemicellulose, and one important component is xylem. Right? Now, we have discussed about all the layers of cell wall, that is primary cell wall, secondary cell wall, and tertiary cell wall if present, that is in gymnosperm, tracheid of gymnosperm. So this complete layer, if we compare this complete layer, this is the inner layer is secondary cell wall, right? This sky blue layer is middle lamella, and this pink layer is primary cell wall. So location is primary cell wall is towards the outer side, right? Location of secondary cell wall is towards the cytoplasm, which is very important. This question may be asked in your part test or maybe in your exam, that secondary cell wall is formed towards the cytoplasm and primary cell wall is present toward the outer side or towards the middle lamella. Now, what is middle lamella? What is the function of middle lamella? If you can see, this middle lamella is present between these two cells, or we can say two neighboring cells. So, function of middle lamella. Middle lamella is cementing material between two neighboring cells, right? Component of middle lamella, chemical composition. It is made up of calcium and magnesium pectate, right? More composition of calcium. If we compare composition of calcium and magnesium, then calcium component is more than magnesium, right? Also known as plant cement, because it helps in joining of two plant cells, right? Next is plasmodesmata. Now, what is plasmodesmata? Suppose we have two plant cells, right? Connected at this end. So these two plant cells are connected through some cytoplasmic bridges for transport. And these are the cytoplasmic bridges. So this connection is plasmodesmata. This is plant cell 1 and this is plant cell 2. So plasmodesmata is nothing but it is cytoplasmic connections between two plant cells, right? Or we can say cytoplasmic bridges between two plant cells. Plasmodesmata term was given by Strasberg in 1901. Here is not important, you just need to remember the name of the scientist. Now, this cytoplasmic bridges or plasmodesmatal connection are made up of, it is made up of ER tubules. ER tubules means it is made up of, ER means endoplasmic reticulum. ER tubules. ER tubules are also known as or desmotubules. This term is very important. This plasmodesmatal connection is made up of ER tubule or desmotubule, which forms the bridge between two cells. And this desmotubules help to maintain continuity of cytoplasm, right? So one important special note which is given in your NCRT, that the cell wall, which is toward the inner side, and middle lamella, which is toward the outer side of the cell. Cell wall and middle lamella may be traversed by plasmodesmata, which connect the cytoplasm of neighboring cells. Now, what do you understand by traversed? Traverse means which travels through, right? So, cell wall and middle lamella travels through plasmodesmata from one cell to the other cell. Next point about cell wall is function of cell wall. Now, what is the function of cell wall? Why cell wall is present in plant cell or fungal cell or bacterial cell? It provides shape to the cell, right? Second, it protects the cell from mechanical damage and infection. It ensures that there should not be any damage or infection to the cell. So it protects the cell from mechanical damage or infection. And it helps in it helps in cell to cell interaction. Means two cells will interact through obviously their outer membrane, which is cell wall. Cell to cell interaction, right? And it provides, since it is outer membrane of the cell, so it provides barrier to undesirable, which is not required by the cell. Undesirable means which is not required by the cell. Macromolecule, there are two type of molecule, macromolecule and micromolecule. If the size of molecules are larger, then it is considered as macromolecule, and if the size of molecules are smaller, then it is considered as micromolecule. So it provide barrier to undesirable macromolecule. Why it is not barrier to micromolecules? But because for micromolecules or general or most of the macromolecules, cell wall is permeable, means it allows the entry and exit of micromolecule, but it is barrier for undesirable macromolecules. This point is very important, right? Next component of the cell is plasma membrane, right? Now, what is plasma membrane? Is it the outermost covering of the cell or innermost covering of the cell? How will you define plasma membrane? First, in animal cells, plasma membrane is considered to be the outermost covering of the cell, right? outermost covering? Because cell wall is absent in animal cell, right? But if we compare this with plant cell, then in plant cell, plasma membrane is present inner to the cell wall. So outer membrane is cell wall. Just inner to the cell wall we have plasma membrane in case of plant cell, or fungal cell, or bacterial cell, or some of the protist. Right? Now, if we compare this structure of plasma membrane with cell wall was non-living and rigid structure. Plasma membrane is living and flexible structure. Plasma membrane of cell is considered to be selectively permeable. This is very, very important. Now, what do you understand by selectively permeable thing? For this, we have to learn about permeability of membranes. Right. Here we will write permeability, right, for solute and solvent. First permeability type is completely permeable. Now, completely permeable means it will allow the entry of solute and entry of solvent, both the molecule. Now, what is solute and what is solvent? Suppose we have a solution of water and sugar. This is a solution of water and sugar particle. So, sugar particles are considered to be solute, while water is considered to be solvent, right? So if any membrane allows the movement of solute as well as solvent, the membrane is considered to be completely permeable. Example of completely permeable membrane is cell wall, right? Second is impermeable membrane. Impermeable or completely impermeable membrane, we'll write completely impermeable membrane. It will not allow either the entry of solute or solvent, right? Next type is semi permeable membrane. Semi permeable membrane will not allow entry of solute, but it will allow entry of solvent. Semi permeable membrane is not present, or we can say it is absent in living system, right? Where it is used? It is used in dialysis machine. Used in dialysis machine. In dialysis machine, the membrane used is semi-permeable type of membrane. Next type is selectively permeable membrane. Now, if we talk about selectively permeable membrane, in selectively permeable membrane, solvent is allowed, solute is also allowed, but only selected solute particles are allowed, right? So this type of membrane is selectively permeable membrane, means it allows the movement of solute, but only selective solute particles are allowed. And example is plasma membrane. So whenever we talk about plasma membrane, we have to consider plasma membrane as selectively permeable membrane. That means it allows the entry of solvent particle, but it allows entry of only selective solute particles, right? Selective solute particles. Now, when was structure of this plasma membrane was studied? Biochemical investigations of detailed structure of cell membrane was done after the advent of electron microscope. Means after the discovery of electron microscope, it was studied. That is in 1950s. Now, which is the best cell to study structure of plasma membrane? It will be RBC, because cell organelles or nucleus are absent in RBC, so it is the best cell to study the structure of plasma membrane, right? In human RBC, 52% in human RBC plasma membrane is made up of 52% component is protein. That is approximate data, right? 52% protein and 40% is lipid. This data is very important for NEET. It has been asked many times that human RBC contains 52% of protein or 40% of lipid, and if they want to make the statement incorrect, then it will be asked like 52% lipid, which is completely incorrect, right? If they're saying 52% lipid, that statement will be incorrect. It is 52% protein and 40% of lipid. Now, there is one important special note. If we compare the structure of plasma membrane of prokaryotes and eukaryotes, then structure of plasma membrane of prokaryotes and eukaryotes are almost similar. The only difference is about the cholesterol particle, which we will discuss in the structure of plasma membrane, right? So in general, structure of plasma membrane of prokaryotes and eukaryotes are almost similar. Now, model which is study for structure of plasma membrane. Which model is studied for structure of plasma membrane? It is fluid mosaic model. Fluid mosaic model. This fluid mosaic model was given by Singer and Nicholson in 1972. It is very important again for NEET point of view. This has been asked many times in NEET. It was given by Singer and Nicholson in 1972. It is the latest and most widely accepted model of plasma membrane. It is latest and most widely accepted model for the structure of plasma membrane, right? Now, what is fluid mosaic model and what are the component of plasma membrane which we will study in fluid mosaic model? First, we'll draw the structure of plasma membrane according to fluid mosaic model. Right. So if we consider this as structure of plasma membrane, so these two layer are considered as phospholipid bilayer. Now, what is phospholipid bilayer? So, first component of the plasma membrane is phospholipid, or we can say lipid. Phospholipid is the main component of the membrane. Main component of the membrane. Why it is considered as main component of the membrane? Because it forms continuous structural frame of cell membrane. In between these phospholipid biomembrane structure, these protein, or we can say integral proteins, are present. Right? Integral protein, or we can say transmembrane protein. Now we'll discuss about the structure of transmembrane protein or integral protein or peripheral protein. But first we'll be discussing about the structure of phospholipid. Now what is phospholipid? Why we are studying phospholipid? What are the main important point of phospholipid? Phospholipid, structure of phospholipid is like this. That is the head of phospholipid. It is tail of phospholipid. This structure is considered to be head of phospholipid, and this structure is considered to be tail of phospholipid, right? Now if we compare head of phospholipid, it is towards the outer side. Now why we are considering it is towards the outer side always in lipid bilayer? Suppose this is lipid bilayer, head is always toward the outer side and tail is always toward the inner side. This will be out of the cell structure medium, or this is inside of the cell medium, which is cytoplasm. So always this head particle is toward the outer side and this tail particle is toward the inner side, and this is always towards the outer side, right? So head of phospholipid is toward the outer side. Now there is a reason why it is always present toward the outer side, because it is polar in nature, or we can say hydrophilic. Hydrophilic molecules or water loving molecules means it can be in the medium of water, right? If we compare tail of phospholipid, this tail structure is made up of, it is rich in unsaturated fatty acid, or which are liquid in nature, right? So unsaturated means which carbon component which are double bond or triple bond, right? So this is made up of lipid is hydrophobic. For hydrophobic molecule we also use a term as non polar. If any chemical or any component is hydrophobic means it cannot be in contact with water, so that component should be towards the inner side of the membrane, right? If it is hydrophilic, that means it can be in contact with water. So always remember this head is towards the outer side and tail is toward the inner side of the bilayer. Understand? So now we will discuss about one important term, that is amphipathic molecule. Now what do you understand by amphipathic molecule? Amphipathic molecule means molecules which have both hydrophilic as well as hydrophobic molecule. So hydrophilic is head, hydrophobic is tail. So example of amphipathic molecule is phospholipid, right? So phospholipid, we can consider that phospholipids are amphipathic molecule because of presence of hydrophobic and hydrophilic component in a single structure. Hydrophobic component is the tail, which is made up of unsaturated fatty acid, and hydrophilic molecule is the polar head, which is toward the outer side of the membrane, right? So that is the structure of phospholipid. Now, next important point about phospholipid. This structure of phospholipid, why phospholipids are present in plasma membrane? Phospholipid layer provide fluidity to the membrane. Now, why it provides fluidity to the membrane? Just now we have discussed that tail is rich in unsaturated fatty acid which are liquid in nature, and that nature provide fluidity to the cell membrane, right? Because of fatty acid tail, right? So we consider plasma membrane to be quasi-fluid in nature because of phospholipid. So quasi-fluid, quasi-fluid means jelly-like, not exactly fluid. Quasi-fluid nature of lipid provide flexible nature to the cell membrane. It enables lateral movement of proteins. Now, what is lateral movement of protein within the overall bilayer? Now, what do you understand by lateral movement of protein? Suppose this is the structure of plasma membrane, right? Fluid mosaic model of plasma membrane. That is a protein. If I rotate the protein about 180 degree, this rotation is the lateral movement, and this lateral movement is provided by the movement of these lipid or fluid or flexible nature of this lipid. If these lipids are always fixed in a position, then this movement would not be possible in that case, right? So, the quasi-fluid nature of lipid enable lateral movement of protein within the overall bilayer. This important is very important, and this ability is known as measured as the fluidity of the membrane. That how much lipids proteins are able to move within the overall bilayer, that means that much fluid is the plasma membrane. Now, why fluid nature of plasma membrane is important, how it will help. So the fluid nature of the membrane, it is important for various function. What are those various functions? Various functions are: number one, cell growth; number two, formation of intercellular junctions; number three, endocytosis. We'll learn about what is endocytosis, complete process of endocytosis. Number three, secretion, and number four is secretion, and number five is cell division. These are given in your NCERT. There are more functions which is performed by plasma membrane on the basis of its fluid nature, but major function which is performed by plasma membrane is cell growth, formation of intercellular junction, endocytosis, secretion, and cell division. These are most important function which is performed by plasma membrane. Next, if we are talking about lipid, there are minute structure which is present in between lipid. These are the structures. So these structures are known as cholesterol particle. Now, why cholesterol particle is present? Cholesterol particles provide stability. Cholesterol is also present in plasma membrane. Why it is present in plasma membrane? Because these cholesterol particles are more rigid than the lipids. Since lipids provide fluidity, so these more rigid particles will provide the stability. So it provides stability to the membrane, right? Now, the only difference between prokaryotic and eukaryotic plasma membrane is presence or absence of cholesterol. This cholesterol is absent in prokaryotes. Why it is absent in prokaryotes? Okay, if cholesterol particle is absent in prokaryotes, then what are the other component which is present in prokaryotes for stability? It is hopanoids or a type of sterol, right? It is present. That name or the term is not very important for your exam point of view, but it is just that cholesterol particle is absent in prokaryote, that you need to remember, right? Now, next, that was all about phospholipid. Next component of plasma membrane. Next component of plasma membrane is lipid, right? Which is, sorry, next component of plasma membrane is protein. Now, protein on the basis of ease of extraction, it is categorized in two categories: integral or intrinsic proteins. Next is peripheral or extrinsic proteins, right? This is categorized on the basis of ease of extraction, right? Now, integral protein means which is tightly binds with phospholipid. It is very difficult to extract the integral protein. It is either partially buried or it is completely buried in the membrane. So if it is completely buried in the membrane, means it is completely penetrating the membrane, it can form a these type of proteins forms a passage across the membrane. Now, which type of passage is formed? Passage in terms of, in the form of channels, or we can say in local language we also use a term tunnels, right? These channels or tunnels are only for movement of water soluble substances or molecules. Now, why water soluble molecule and why not fat soluble molecule? If we consider the structure of plasma membrane, suppose this is the structure of plasma membrane. This is the structure of plasma membrane. This region of plasma membrane is considered to be hydrophobic, means water loving molecule or water soluble molecule directly through diffusion cannot cross this plasma membrane. So, for these molecules, we need some specialized channels or protein channels or tunnels, and those Protein channels or tunnels are made by integral protein. Simple. For the movement of water soluble molecule, right? Next is peripheral or extrinsic protein. Peripheral or extrinsic protein means it is present superficially. Since it is arranged superficially, that means extraction is very easy, right? It can be extracted easily, right? Why these proteins are present? These have some enzyme activity, enzymatic activity. For that purpose, those superficial or extrinsic proteins are present. Now, we have discussed about protein, structure of protein and structure of lipid in plasma membrane. Is there any other component which is present in plasma membrane apart from lipids and proteins? Yes. Those molecules are oligosaccharides, or we can say sugars, right? These oligosaccharides are of glycoproteins or glycolipids. Now, what are glycoproteins or glycolipids? Glyco means carbohydrate component and protein means protein. When carbohydrate is attached to the protein group, then it is glycoprotein, and if it is attached to lipid group, then it is glycolipids, right? It is present on outer surface of plasma membrane. What is the function of these oligosaccharide particles? It is helpful in cell to cell recognition. Now, the most famous example of these oligosaccharide are blood antigens. Blood antigens are the example of these oligosaccharide, right, which helps in cell to cell interaction or recognition, right? So, that is the structure of plasma membrane, right? We have discussed all the component of plasma membrane. Next topic of plasma membrane will be transport through plasma membrane. It is a very small topic. We'll finish this topic, and in next lecture or upcoming lecture we'll be discussing about endomembrane system. Four organelles are included in endomembrane system. So, transport across plasma membrane. It can be molecular transport or it can be bulk transport. When more molecules are transported across the plasma membrane, it is termed as bulk transport. In bulk transport, first type is exocytosis and second is endocytosis. We'll discuss about bulk transport later on. First, we'll be discussing about molecular transport. In molecular transport, we have two categories. One is passive transport of molecules across plasma membrane, and the other is active transport of molecule, right? Now, what do you understand by passive transport or active transport? Passive transport means energy is not required. Energy is not required. Energy means in the form of ATP molecule. ATP is not required for the movement of any molecule, then that type of movement is active passive transport. And in active transport, energy in the form of ATP is required, right? So, movement of solute particle in passive transport is movement. Since energy is not required, then movement is also always higher concentration to lower concentration. Suppose we have this system separated by a membrane, right? More solute particle inside A and less solute particle inside B. If the movement is from A to B, and suppose the movement is from B to A. A to B means higher concentration of solute at A side and lower concentration of solute at B side. So this type of movement will be passive movement. And this type of movement is generally we consider it active movement. So here the movement is from lower concentration to higher concentration. For such movement, we require pumps. Requirements of pumps. Example of these pumps are sodium, potassium pump. You will learn about this sodium potassium pump in your human physiology chapter, right? So sodium potassium pump is an example of active transport. Now we'll learn about passive transport, type of passive transport. In type of passive transport, first type of transport is simple diffusion. Second type of transport is facilitated diffusion, and third type of transport is osmosis, right? In simple diffusion, simply solute molecules move from higher concentration to lower concentration without any protein channels. There is no requirement of protein channels for simple diffusion. Example, very important, neutral solutes may move across plasma membrane through simple diffusion. Right? It is very important. Neutral solute may across means they do not have any charge. So neutral solutes may transport across plasma membrane through simple diffusion. Similarly, if any solute particles move from same higher concentration to lower concentration, but it requires with the help of protein channels, then that type of diffusion or transport is facilitated diffusion. Example of very important example of facilitated diffusion you will learn in ATP synthase particle. In ATP synthase particle, F naught channel of ATP synthase. You will learn about F naught channel of ATP synthase in respiration chapter. So that is the example of facilitated diffusion, in which movement of solid particle is from higher concentration to lower concentration, but still it requires some protein channel for the movement, right? Less concentration and more concentration. Movement is still from higher concentration to lower concentration, but since it is requiring protein channels, the type of diffusion is facilitated diffusion. If we talk about osmosis, then osmosis is movement of water molecule from its higher concentration to its lower concentration. Now, what does it mean? Suppose we have a system separated by a membrane. Concentration of solute is higher towards side A and it is lower towards side B. Then concentration of water is higher in B section and concentration of water is lower in A section. So movement of osmosis will be from B to A. That is movement of osmosis or direction of osmosis. These are molecular movement. Number 1, facilitated simple diffusion. Number 2, facilitated diffusion. Number 3, osmosis. In simple diffusion or facilitated diffusion, movement of solute take place, while in osmosis only water molecule take place. So osmosis is a special type of diffusion in which only water molecule move across the membrane, right? Next is bulk transport. In bulk transport, we have two type of transport system. Endocytosis and exocytosis. Suppose that is the cell membrane and foreign particle is toward the outer side of the cell. There will be a vacuole formed by the membrane and food particle is engulfed by the cell, and this type of movement is endocytosis or transport is endocytosis. Suppose this foreign particle, if these particles are, it can be solid or liquid. On this basis, there are two type of endocytosis. One is phagocytosis and another is pinocytosis. Suppose these foreign particles are solid, then it is phagocytosis. If it is liquid, then it is pinocytosis. Pinocytosis is also known as cell drinking, while phagocytosis is also known as cell eating because particles are solid. So definition of phagocytosis is ingestion of solid complex material by membranes in the form of vesicles or phagosome, the process of phagocytosis. Pinocytosis is ingestion of liquid material by plasma lemma. Plasma lemma is the membrane, outer membrane, plasma membrane, in the form of vesicles. So vesicles in phagocytosis is phagosome, and these vesicles in pinocytosis is pinosome. Right? The process is same. While exocytosis is also known as cell vomiting. Cell vomiting means it is egestion or removal of waste material which is not required by the cell from cell through plasma membrane. That is exocytosis, right? That is movement across the membrane. Molecular movement. In molecular movement we have passive transport, active transport, and in passive transport there is no requirement of energy and movement is from higher concentration to lower concentration. Active transport, energy is required, movement is from low concentration to high concentration, and there is requirement of pump. Example of active transport is sodium potassium pump. We have three type of passive transport: simple diffusion, facilitated diffusion, and osmosis. If diffusion of molecule, right, if diffusion of molecule is from higher concentration to lower concentration but without any protein channel, that is simple diffusion. Example आएगा इसमें neutral solute, sorry. And second is facilitated diffusion. In facilitated diffusion, movement is same, solid particle move from higher concentration to lower concentration but with the help of protein channels. So that is facilitated diffusion. It is facilitated by protein channels. In this, there is simple type of diffusion where movement of water molecule take place from higher concentration to lower concentration. So in all three type, movement is from higher concentration to lower concentration. Higher to lower, right? That is transport across membrane. In next lecture we will be discussing about endomembrane system, that is endoplasmic reticulum, Golgi apparatus, vacuole and lysosome.